Yeah.
All right. Okay, please switch on your videos, everyone. So in the previous class, I had told you that there are three stages any neuron could be present in, right? I told you about polarized state, I told you about depolarized state and hyperpolarized. Today I will teach you what these stages are and what do they actually mean. Now, the first case, we're going to talk about the first one, that is polarized state. Remember I told you how poles are formed over here? Now we're going to discuss it in depth. Switch on your videos, both of you. Now, if right here is the membrane of the axon, okay? This outside is the extracellular space. Okay, extracellular space. And inside we call it as the intracellular space. Okay. And this basically is what? It is an axon. Okay. Now what happens during the polarized state when there is no charge, present in the, you can say neuron membrane, there's no impulse, nothing. It's just a simple neuron without any impulse, without any signal. Okay, that's the case. So in that case, outside, the concentration of sodium ion is more. What is the charge carried by sodium ion?
Positive.
Positive charge. So outside, positive charge is more. Okay. And inside, there are more potassium ions. So if potassium ions are there, we can say that positive charge is there. But they in the intracellular space, as compared to the potassium ions, there are many negatively charged proteins present. Many. The things that you can never count upon. Clear? So that is why the all in all, you can say charge of the membrane is in such a way that the outside of the membrane is positively charged and the inside of the membrane is negatively charged. This is the state
But what are
called
these
as the
negative charges?
protein. Proteins that
Oh,
are
okay.
much more in number as compared to the potassium ions inside. That is why they override the charge of the potassium ions. Clear? So this state is what we call as the polarized state. And during this, the potential across the membrane of the axon is minus 70 millivolts. This is the normal resting potential. Clear? This much is clear to all of you, to both of you.
Yes, ma'am.
Okay. Second situation is your depolarized state. Okay, depolarized state. Now, this state occurs when a particular signal arrives at the neuron. This thing, the axonal membrane or neuron in general. Okay, so this when is it? When a signal. See, I say signal, but we shouldn't say signal. We must say impulse. Okay, so when the impulse arrives, so then that state of the neuron is called as the depolarized state. And how does it look like? See. See, normally, if you think about the polarized state, in this ideal situation, remember one thing, the sodium from the outside can't come in, the negative charges from the inside can't go out. Okay, there's no exchange in that matter. Okay. Now, when we talk about the depolarized state, in this state, what happens is that because of the arrival of the impulse doesn't arrive throughout the length of the neuron. It arrives part by part. Okay. It arrives part by part, like taking certain sections and then moving forward. So, for example, if this is the membrane of the neuron. So as soon as the impulse arises, there is a certain channel that opens up which is called as sodium channel. Okay, it's like a gate that opens up. As a result of which, what happens is the sodium from the outside, the sodium ions that were there from the outside, they will move inside. But this is the part where the impulse has taken place. Okay. Furthermore, the impulse is not there. So here the outside will be positive, just like how it used to be, and the insides will be negative, just like how it used to be. But now, in this part of the neuron, the sodium is coming inside, so positive charge is appearing. Potassium itself has positive charge, and then there are negative charge proteins. This is the situation. This is called as a depolarized state. Clear?
Yes.
The third stage or the third stage is the hyperpolarized. Hyperpolarized. Now this is the stage wherein now to balance out. Okay, what it does is that. Acha, one more thing. During this, the sodium comes in and the potassium goes out. Okay. Whereas when we talk about the hyperpolarized state, now during this stage, we have to manage the polarity back again. So 3 sodium from the inside, it is transported out. Clear? To manage the polarity. So that is called as
Ma'am,
the
what do you mean 3 sodium?
3 sodium meaning out of the total amount of sodium that is there, few of it is pumped back into the extracellular space. When I read the language, you people will understand it much better. Resting potential. See, when the neuron is at rest. So when the charge separation across the axonal membrane is maintained, the neuron is said to be at rest. Meaning when outside there is positive, inside there is negative, we say that the neuron is at rest. And the potential that we get is called as the resting potential. Okay. A resting neuron is negatively charged on the inside and it is positively charged on the outside. Such a charge separation is called as polarization, and so we call the neuron to be polarized. The first situation that we discuss. Okay, we'll say that it is neuron at rest. Clear? Any doubts?
No, ma'am. No, ma'am.
Okay. Now, at the resting potential, the membrane of the axon, called as the axonal membrane, it is more permeable to potassium ions and it is impermeable to sodium ions, meaning sodium ions cannot come inside. Potassium ions can come. And from the inside, proteins can't go outside also. Okay. Similarly, the membrane is impermeable to negatively charged proteins, like I told you. Right? That is present in the exoplasm, meaning in the intracellular part, the proteins that are there, they like the membrane is impermeable. Neither can it go out nor can it come in. Clear? Because these are bigger molecules. Understood?
Yes. Yeah, ma'am.
The exoplasm contains high concentration of potassium and negatively charged proteins and low concentration of sodium, almost negligible concentration of sodium inside. Whereas outside there is a high concentration of sodium and low concentration of potassium. This creates a gradient, high to low gradient, isn't it? So now, remember I told you how during the hyperpolarized state, the sodium is pumped outside so that the potassium could be brought back inside. Isn't it? This is the case. So it is written over here that these ionic gradients across the resting membrane are maintained by active transport by the sodium-potassium pump, which transports 3 sodium outside and takes 2 potassium inside by spending energy. So this right here, during the hyperpolarized state, this is called as the sodium-potassium pump. So you are using pump and it requires ATP. So you're using pump and it requires ATP. So with the expenditure of ATP, you're sending 3 sodium ions outside, and you're taking 2 potassium ions inside. Clear?
Yeah, ma'am.
So that's the case. So thus the outer surface of the axonal membrane, now it possesses a positive charge, and the inner surface becomes negatively charged again, back to how it used to be. Now the action potential, the time when the nerve impulse gets initiated or gets generated. Okay. So the transmission of a nerve impulse, called as the action potential, to differentiate it from the resting potential, it occurs in the 4 phases. See, the reason why we're calling it as action potential, so that you don't confuse it with resting. Resting potential is when no signal, no impulse is at the neuron. Action potential when the impulse has now reached the neuron. First, initiation of an action potential by sufficiently large stimulus called a threshold stimulus. Okay. Whenever our neuron has to be stimulated, this thing, a stimulus has to be provided. Stimulus could be internal, stimulus could be external. Stimulus
Ma'am, your
means,
voice is not clear.
is
It's
it
low.
okay now? Is it okay now? I was saying that whenever a nerve impulse has to be generated, during that time, a stimulus has to be sent. Stimulus can be sent from the inside of the body also and from the outside also. For example, if someone touches your hand, that touch is a stimulus. The stimulus is the information or sort of like the change in your environment, either inside or outside. Clear? Then that stimulus sends or it gets transmitted along the entire length of the neuron, because that's the unit of our nervous system. Then it transfers or it's transferred to a target cell, which can be a neuron or a muscle, depending upon what, like for example, if um, I'm touching your hand with something very cold during winter season. So what are you going to do? You're going to pull your hand back. Ma'am, are you crazy? Pull your hand back, right? So this pulling thing, your muscles are involved, and this signal that you've received is because of your neuron. Okay, so now the target cell shows the effect. This is clear. The basic steps is how to initiate a nerve impulse.
Yeah.
Okay. Action potential. Now, this is very important. Remember that the action potentials, they are all or none phenomena. All or none means either the stimulus will be provided and you'll, like the whole entire nerve impulse will happen. Or the opposite, it won't be provided and the nerve impulse won't happen. But you can't do half of it, like half of stimulus is provided, partial stimulus. No, stimulus is always full. So that is what we call as all or none phenomena. They do not occur without sufficient stimulus. Okay, if the stimulus is not sufficient, nerve impulse conduction will not happen. But if they have started, then it cannot be stopped. It's not something that you can stop in the middle. It's not something that you can change in the middle. So it's all or none phenomenon. Okay. Every action potential causes the same amount of voltage change in the neuron's interior. There are no large or small action potential, only large and small stimuli. Stimuli meaning if someone is just touching you like this, that is a stimulus. If someone is putting on 10 kgs of weight on your hand, that is a large stimulus. Understood? It could be anything. If a very fat person comes and sits on your hand, that is a different kind of stimulus. Understood? So the strength of the stimulus can vary, but the potential that, like, if your a very small stimulus is applied, then also the same action potential will take place. No potential change will happen in the membrane of the axon. Even if no matter how big the stimulus, even if something very heavy falls on your hand, then also the same potential is there. Potential, you people have studied in chemistry. Potential. So you know it better than me. right? Potential and stuff, I don't know. But you know that it is calculated in volts and stuff, right? You know that much. है ना? This much is clear to everyone. Any
Yeah.
doubts?
No, ma'am.
Any confusion?
No, ma'am.
I'm taking this very slow because this is the only part that you'll get stuck on. This is the only part you'll find very weird and confusing. Okay. Action potential temporarily disrupt the polarization of a neuron, resulting in depolarization. Remember when I told you how when the signal or the impulse will be received, the sodium pumps will open, thereby bringing sodium inside the membrane of the axon, right? This is the only state when the neuron is not going to be in the resting potential, meaning in the polarized state. Okay? Why? Because the impulse has arrived over there. Getting me? So action potentials temporarily disrupt, meaning disturb the polarization of a neuron, resulting in the depolarization. When a stimulus is applied to a site on the neuron, the voltage gated sodium channels, they suddenly open and it allows the sodium ions to enter inside the neuron. The net influx of the sodium ion far exceeds the movement via leak channels and sodium potassium pumps, meaning a lot of sodium ions come inside during the time of impulse being provided to the axon or to the neuron. Okay, this sudden movement of positive ions into the neuron cause the neuron's interior to develop a positive charge. Initially, the interior of the neuron had negative charge, but now, since the sodium has entered inside the neuron, it has made it to become positive in its charge. Clear? Next, the voltage gated sodium channels, they will close, and then potassium channels will open, in which the potassium ions, they are sent out. Okay? Remember, we studied how the sodium will come in, potassium will go out. This state is your depolarized state or repolarized state. Clear? Understood? And now we come on to the last stage, that is hyperpolarization. Hyperpolarization is again that stage wherein 3 sodium will be pumped out and 2 potassium will be sent inside so as to maintain back the intake or the potential. So generation of action potential is clear to both of you.
Yeah,
Yes.
ma'am.
Any doubts?
No, ma'am.
So this was about the potential and the changes. So see here, the resting state, the first stage wherein the potassium diffuses more rapidly and potassium can go out, come inside, it's all the case, but sodium can't come in. Whereas during the other states, sodium comes. Now, remember I told you that how the impulse, it's only applied to a certain area. Do you see this shaded area? This is only till where the impulse is. Then when it has to travel to the entire length of the neuron, then it will move here and become this part. Then it will move here and become this part. It doesn't get transmitted thoroughly throughout the length of the neuron as it is. Okay, it goes part by part or it sort of jumps. Clear? Hmm?
Yes. Yeah, ma'am.
Anything you all want to ask?
Ma'am, does the impulse come in through the nodes of Ranveer?
Oh, dendrites.
Oh, so then how does it go part by part?
It's not for that, dear. See, it's throughout the length of, you can say, these jumping, we can consider it as through the nodes of Ranveer. But the impulse will come from the dendrites only, nowhere else.
So the
Dendrites are
jumping is the
uh,
nodes of Ranveer, right?
throughout
Okay.
the axonal length and everything. But mostly, the impulse will come via the dendrites. Clear?
Yeah, ma'am. Thank you.
Okay, so tell me, what is the last thing you people have written? Then we'll talk about this nerve impulse propagation. Last thing you people have written is?
My notes are all scattered. I have screenshots.
From this chapter, you've not written it.
Yeah, because I missed the first part.
Zainab, can you tell me what is the last thing you've written?
Yes, ma'am.
I believe you people were writing this part. This myelin thing was completed in the
Yes, ma'am,
previous.
completed till here. This even the down little short note too.
Stages also you people have written.
We have to start from there.
Aisha, from here onwards you write, okay? The rest of the parts I'll send you the notes for, but from here onwards you note down, okay?
Okay, ma'am. I will.
That
Done, mom.
Both of you are done?
yes, ma'am.
Both of you are done?
Yeah, ma'am.
And then
Both of you are done till here? Yes, both of you are done till here? Aisha, are you done or not? Can you hear me, students?
Yeah, ma'am.
Done till here?
Yeah, ma'am.
Okay. Now let us move forward and see how the process of the nerve impulse being sent or the propagation takes place. Now, when we are talking about the propagation process, in this we're going to see how the whole entire nerve impulse gets from one point in the, you can say nerve or the axonal membrane to the next point. This is what we have to study in this particular topic. So when we talk about the propagation or conduction of nerve impulse, the action potential or the nerve impulse, in contrast with the resting potential, it propagates along the membrane from one point of the neuron to the farthest extension of the neuron. The resting potential does not propagate, while the action potential, once developed, it will propagate to the farthest extension of the neuron. Remember, resting potential stays just the way it is and it's not going to move forward. Nothing is going to happen to it. Neither will it move forward, nor will it move backwards. Nothing will happen. Whereas when we talk about the particular action potential, once it is generated, it has to be transferred throughout the entire length of the axon. Now let us see how that happens. Now here we see that there are sites marked like A, B, then C, D and E. Likewise. So at the sites immediately ahead of the site A on the axon, like site B, the axon membrane has a positive charge on the outside and negative on the inside. Why? Because in this condition, since the impulse has only reached the first stage, the B portion of the neuron will be in the normal polarized state. Okay, but side A has positive charge and the B has negative charge on the inner side. The current positively charged, like positive charge, flows from the outer surface from side B to side A to complete the circuit of the current. Because you people know that it will always move towards the opposite charge direction. Hence, the polarity gets reversed and an action potential gets generated at the site B. So why does the action potential from site A and it gets generated at the site B? Basically, because we know that on the outer side, the site A has negative charge. Okay, because at that point of time, the sodium is inside and the potassium is outside, or the opposite case scenario is there. So, and the site B has positively charged. So, the current flows from site B to site A. This changes the polarity. Thereby, the action potential gets generated at the site B. And same way, it will keep on generating for however many sites will be present on the axon. Is it clear, students?
Yeah, ma'am.
So for the entire length of the axon, this thing keeps on happening. Thereby, the nerve impulse gets transmitted from site A to B to C to further more. Okay, so this is what we call as the propagation or the moving forward of the nerve impulses that are there. Is it clear? And this whole process is extremely fast. We are studying it slowly because for the sake of understanding, but this depolarization, repolarization process is very fast and usually it takes around 1 to 5 milliseconds. Imagine how quick it would be. Is it clear, students? If people
Yes,
have
ma'am.
any doubts up until now?
No, ma'am.
No doubts? Okay. Now, the next thing we're going to talk about is the transmission of impulses. Now, you people know, up until now, we have studied how the impulse comes to the neuron and generates an action potential and everything, right? But how does it get transmitted from one neuron to another neuron? And do you people remember the space that is present between the two neurons? What is it called as?
Synapse.
Yes, synapse. Synapse is present. It is basically a gap that is present in between the two neurons, meaning the axon ending of one neuron and the dendrites of another neuron, right? That is what we call as synapse. It's like a junction. It's like a space that is present. Clear, students?
Yes, ma'am.
Now let us see how that would happen. For example, if we are thinking about the nerve impulse transmission from one neuron to another and furthermore, so it could be, the next topic we are discussing is transmission of impulse. Transmission of impulses. Now, how does that happen? For example, here, right in the middle, we have our neuron. The neuron number 1 is over. All its dendrites and everything, the neuron is. Now, I am saying that always the connection between the neurons and like neuron number 1 and neuron number 2 is going to be like the neuron, like the nerve endings of one neuron and the dendrites of another. So, for example, if I am to connect, now it could be connected from any side. Okay, it's not that it could be connected in vertical section. It could be connected in any way possible. So, for example, if this is the area, then what's going to happen is that this is the dendrite portion. And then now we're going to start with another neuron, which is over here. So for example, if this is another neuron, which is here. So the nerve endings of this neuron is basically present towards the dendrites of this neuron, right? This space in between them is what we call as a synapse. Is it clear? This space is the synapse. Now, if the impulse, okay, if the impulse is moving from this neuron that I'm drawing, so if this is neuron number 1, if this is neuron number 1 and this is neuron number 2. So if the impulse is moving from the neuron number 1 to the neuron number 2, then this area will be called as the presynaptic membrane. Presynaptic membrane, meaning much before the synapse arises. Then we have the synapse, and then the part that will be present over here will be called as the postsynaptic membrane. You people are following me? Understanding everything?
Yes,
Yeah.
ma'am.
Good. So that is your postsynaptic membrane. So this is where the whole entire neuron transmission will take place. Now, what are these things we must talk about? So the first one is a synapse. What is a synapse? It is basically the gap, okay, present in between two neurons. Okay, so we call it that. It is the gap present between the nerve endings or neural endings of one neuron and the dendrites of another. Right? This is the gap wherein we call it as synapse. Okay. Now, when we talk about the synapse, one more thing is that it is formed by the membranes of the presynaptic neuron and the postsynaptic, isn't it? So we'll write that also, that it is formed from the membranes of presynaptic neuron and postsynaptic neuron. Is it clear?
Yes, ma'am.
Yeah, ma'am.
Okay, very good. Now, furthermore, when we have discussed this, then we must know that what is a presynaptic neuron and what is a postsynaptic neuron in proper sense. So, when we talk about the presynaptic neuron, so this is the part that is basically present before the synaptic gap or the synaptic cleft. Okay, the part present before the synapse is called as presynaptic neuron. The part that is present after the synapse, we call it as the postsynaptic neuron. Clear?
Yes, ma'am.
Yeah, ma'am.
See, mostly when we talk about the synapse, we also simultaneously measure the synaptic cleft. See, actually, the main thing, the synapse is the space between, or many a times we call it as the synaptic gap or the synaptic cleft. Synaptic cleft or the gap. The actual gap that we were talking about, okay, it is present between the presynaptic membrane and postsynaptic membrane, and it is usually in terms of nanometers. So it is 10 to 20 nanometers wide. Okay, it is 10 to 20 nanometer wide. Clear, students?
Yes, ma'am.
Any doubts up until now?
No, ma'am.
No, ma'am.
All right. So we'll write one line for the presynaptic membrane also. It is the membrane or it is the part of neuron present before the synapse. The part that is present before the synapse is what we're going to call as the presynaptic membrane, and the part that is present after the synapse, we're going to call it as postsynaptic membrane. So it is the part okay. Now, further, when we have discussed about this, we must also discuss about the different types of synapse that are. Okay, this is also important. The types of synapse. Now, when we talk about the different types of synapse, we basically have two that we have to discuss. Okay, the first is electrical synapse. And the second is your chemical synapse. Is it clear, students?
Yes, ma'am.
Yeah, ma'am.
All right. So talking about the electrical synapse here, the nerve impulse, it basically gets conducted via electrical signals. Okay, and here the pre and the post synaptic neurons, they're very close together. Okay, they're like the space between them, the gap or the cleft between them, it's very, very less. So they're very closely situated to each other. Chemical synapse, on the other hand, this is the one in which the gap will be more and the gap will be containing a certain fluid present in between. And this is regulated with the help of a chemical called as neurotransmitter. Remember acetylcholine, dopamine, all of that?
Yes.
So it gets regulated via that. Like people note down this ion channels. This part is done. The axonal membrane, the polarized state is done.
Yeah.
Yes, ma'am.
All right, students, done till here?
Yes, ma'am.